Hello, this is Divya. I'm doing my elective assignment on metabarcoding. I feel like this is a very interesting topic as the first time I've heard of metabarcoding was in this class. So first, what is DNA? DNA, also known as deoxyribonucleic acid, is like the instruction manual for all life. It's made of 4 different nucleotides, which you can see over here: adenine, thymine, guanine, and cytosine, represented by A, T, G, and C, which are like the words in the construction manual called DNA. The DNA, once read, can be used to make proteins, which are like the screws and the woods to make a furniture, which is going to be the organisms of us. Then going on to eDNA specifically, which is the DNA that many organisms leave behind in their environment, hence the E. It can be found in land, water, practically anywhere an animal goes. The sources of DNA can be from like excretions, skin cells, etc. eDNA analysis, which I'll call metabarcoding, is a non-invasive way to get DNA of organisms without causing major disturbance to the environment or the organisms themselves. And metabarcoding, the process itself can be divided into 3 main sections, which is collecting samples, doing a wet lab, and then a dry lab. Collecting samples is as simple as it sounds. We go to the environment and take samples of the environment where we think, oh, there could be a species here. It'd be like soil samples, sediment samples, etc. After that, we go into the wet lab portion. So first, we have this sample. It has all the soil, and in the soil, we should be able to find some cells that have DNA of organisms, hopefully. So first we need to separate the DNA from the rest of the cells and the rest of all the other junk that was collected. So first, we lyse the cell, we break it down either manually or with a chemical. You can use a centrifuge or some other method to separate the DNA so it's separate from the rest of like the cells in our matter. Then we take that DNA out, put it in its own container, and we move on to a step called PCR amplification. In PCR amplification, we look for certain sections of nucleotides that tell us, oh, this is a very good gene to choose to look at. And then we amplify these segments. In other words, make many more of these. A good segment of DNA is one that's not very long or very short, so that we'll be able to have enough DNA to analyze, but not so much that the computer overloads. In addition, the DNA should have high interspecific variation, which means that there's a high genetic variation of this gene between different species. And it should have a low intraspecific variation, which means that organisms of the same species should not have much variation between the nucleotide sequence for that gene. The last part of the wet lab is the library preparation. So after we amplified the sections of DNA, we have to take out all the unused sections of DNA and any PCR DNA fragment remains. And then we have to tag the different amplified samples so we can differentiate them. Because of course, we're not only looking at one specific part of a gene out of all the DNA we have, we're going to be choosing many different little segments of many different parts of the DNA. So we want to have an equal amount of all of these samples that we collected. So we'll be using equimolar amounts to create this final solution. The reason we need an equimolar amount is so we don't overlook any of the other organisms that are in the area. Say there is very little of one species and a lot of different species in that specific area, but we want to be able to catch all the organisms, so we're going to use equal amounts of DNA from both. After the wet lab, we go to the dry lab, which is the quote-unquote computer portion, also known as bioinformatics. Here, we look at these amplified segments, separate them because they were tagged during the wet lab portion, so we can separate each segment into the different groups of DNA samples that were amplified. Then we compared these nucleotide sequences to a taxonomy table to see any matches or differences in the eDNA sample with scientists that previously identified as the DNA sequence for a species. And then the general application for this process. So the species identified can be used to see patterns of the population of species in an area, like, oh, this species is here, and it can also be found here. This way, we can monitor biodiversity and have a better understanding of all the organisms in our environment. In addition, due to climate change and pollution, many biomes are lost, which changes the ecosystem and organisms in it. eDNA analysis can help in documenting these changes as they occur. Furthermore, as stated before, it's a less invasive way of identifying all the species in the area, so we're much less likely to affect the ecosystem just by going into it, and it's more likely that it's due to the climate change and all the pollution in the area. Furthermore, one other great thing about the DNA metabarcoding process is that it's easier to detect rare and elusive organisms by their remains instead of having to actually go out, find these organisms, and then take a sample from them. This gives us a more holistic view of the organism in the area as it's much easier to detect them. And from all of this information and all of these applications, it makes it much easier to predict the changes of the biodiversity in the area and how that will affect us in general. These are my images citations, and I hope you have a good day.